Hello, my name is Shambhavi Gautam, and I am a sophomore studying bioengineering. Over the summer, through the Penn Undergraduate Research Mentorship Program, I studied in the Zhou lab and conducted research on CDKL5 deficiency disorder. This presentation will dive into some of the insights I found into the pathophysiology of CDD. In this presentation, I will give a brief introduction of what CDD is, the mouse models we use to study it, and specifically diving into interneuron development in my project. And finally, I will go over conclusion and further steps in my lab. So what is CDD and how is it different from other disorders in the brain? First and foremost, CDD stands for cyclin-dependent kinase-like 5 deficiency disorder. It is a monogenetic disorder, meaning a genetic disorder caused by a mutation in a single gene under the umbrella of autism spectrum disorders. But it is different because it is caused by a singular gene being disrupted. Its phenotypes are similar in terms of hypotonia, developmental delays, intellectual disabilities, visual impairment, and most important for this project, early onset epilepsy in infants, which we will investigate in our project. So what is CDKL5? CDD is distinct because it is caused by an N-terminus mutation in CDKL5. It is an X-linked gene encoding serine threonine kinase 9. It has two domains, with most pathogenic mutations occurring in the N-terminus because this is the part of the protein that directly interacts with substrates. So mutations are more prone to cause problems. More specifically, mutations can cause issues in neuronal migration, axonal outgrowth, dendritic morphogenesis, and the development of synapses. Since we know what causes CDD, how do we study CDKL5? In order to most closely resemble human development and growth, we chose a mouse model so we can correlate genetic changes with behavior and have relevance in our field where most labs utilize mice models. We're able to create mouse models that resemble human development and growth, as well as similarly represent the effects of the genetic mutation in humans and in mice. There are many mechanisms available to facilitate genetic manipulation, but in our lab, we use the Cre-LoxP system. This is comprised of Cre, a site-specific recombinase, and LoxP, which is a DNA sequence made up of two recognition sites, as well as a spacer site in the middle. When DNA is floxed in the same direction, the site-specific recombinase will excise the DNA fragment. By excising exon 6, we are able to study how CDKL5 affects brain behavior. In addition, we are able to create a conditional knockout model by flanking exon 6 using the Cre-LoxP system. And Cre allows us to target specifically excitatory neurons in the forebrain. In this type of model, we can control where and when the gene is knocked out. This means that in the targeted tissue, the gene of interest has no expression, whereas the rest of the body, the gene is functional but floxed, frequently allowing for spatial control. So in order to do this spatial control, we use NEX, which is a specific gene control in targeted cells. NEX-Cre encodes glutaminergic neurons. In this project, we are focused on interneurons because CDD's most prominent phenotype, early onset seizures, is thought to be caused by unequal firing of excitatory and inhibitory neurons. Interneurons are GABAergic or inhibitory neurons making up 20% of all neurons. By manipulating excitatory neurons, we study the downstream consequences in inhibitory neuron expression. Our staining data utilizes fluorescent antibodies that act as interneuron markers to observe changes between conditional knockout and wild type, as well as early and late rescue. Bouncing off of understanding interneurons, we have to understand how they change over mouse development. Most importantly, we see that programmed cell death occurs between P7 and P14. This means around 30 to 40% of interneurons undergo apoptosis. This is important for when we inject the mice to express Cre. Early rescue schedules for before programmed cell death, P3 through P5, and late rescue is scheduled after programmed cell death. Looking at the coronal sections throughout development supports this. As we see, somatostatin positive cells have a significant decrease between P0 and P14. Somatostatin is a type of interneuron that we'll be studying in this project. When we compare the cells that undergo programmed cell death, we see that knocking out CDKL5 exon 6 decreases programmed cell death in SST-positive interneurons. This is shown in the images where you see more neurons in the ZS green image, specifically because there is less cell death. ZS green is a green fluorescent protein we use to mark interneurons. Furthermore, when we look at ZS green of SST-positive interneurons, there is no significant difference between SST-Cre and SST conditional knockout. So we know CDKL5 plays a role in programmed cell death and migration, but when? Can it be rescued? Will conditional knockout or rescue of CDKL5 in excitatory neurons lead to changes in interneuron density? We utilized the two mice models, Flox and Flex, to conditionally knock out or restore CDKL5 in mice in early and later stages using injections. We then collected tissue for wild type, Flox and Flex mice to count the interneuron density. In order to rescue CDKL5 in the mice models, we can use inducible gene expression where the Flex switch will only flip when the inducer tamoxifen is present. Cre is linked to an estrogen receptor and a heat shock protein. When tamoxifen is injected, it enters the cytoplasm of the cell and removes the heat shock protein and allows the Cre-ER system to enter the cell and flex the targeted gene of interest. When performing staining, we see that both early rescue 4-OHT injections and late rescue TAM injections are successful. Building off of the initial data on the previous slide, the stainings of the hippocampus show that there is significant difference in the top left images where early conditional knockout has a greater density than late conditional knockout. This supports our previous hypothesis that the brain increases interneuron numbers to account for the knockout. In the bottom half, we see that there is a significant increase in the late conditional rescue. This can be seen by looking at the hippocampus layers of the wild type and the conditional knockout and the wild type and the conditional rescue. In addition to testing the efficiency of tamoxifen, for our later projects, we will insert DREAD using plasmid AAV vectors to genetically manipulate the cell excitation both early at P0 and late at P21. DREADs, or designer receptor activated only by designer drugs, are chemogenetic proteins that are engineered to respond to a synthetic ligand instead of their original ligand to activate their G protein coupled receptor pathways, allowing us to control nerve cell activity. In this case, we use HM3DQ and HM4DI DREADs for this experiment, which are modified forms of the human M3 or M4 muscarinic receptors, respectively, and CNO as a synthetic ligand as it can cross the blood-brain barrier. In order to deliver DREAD, we use an AAV plasmid, which is a human parvovirus. Recombinant AAV vectors are a favorable method of gene delivery because they can transfect various cell types and induce gene expression in vivo without integrating into the host genome and causing disease. M-cherry is a red fluorescent protein tag and absorbs light between 540 and 590 nanometers and emits light in the range of 550 and 650 nanometers. We will be using this DREAD system to further our understanding of interneuron development. And finally, thank you so much to Zhou Lab and especially my mentor, Jack, for supporting me through this project and teaching me all the skills I need to know in the lab.